Hello everyone, I'm Salman Ibn Yunus. This is the second video of my Bioinformatics with Python tutorial series. In this video, I will talk about biological data. I will give an introduction to biological data. Before that, I would like to give a quick recap about basic biology and some definitions which you need to know before we move ahead of this tutorial series. So first of all, we need to know what is DNA or deoxyribonucleic acid. So most of you might know that this is a molecule that carries genetic information to build a cell and to keep the cell functioning. It is a polymer composed of four nucleic acid units called nucleotides or base pair. Similarly, we have RNA, which is ribonucleic acid, which converts genetic information from DNA into proteins. Then we have proteins. Proteins are complex molecules that perform various functions in cells, which are encoded by genes. So, what are genes? Genes are segments of DNA that code for proteins. So, DNA and RNA are made up of nucleic acids. Nucleic acids encode and express the genetic code that is within the cell. Nucleotides have similar chemical composition but can be distinguished by the respective nitrogenous bases, which are there are four nitrogenous bases: adenine, guanine, thymine, or cytosine. Then we have amino acids. Amino acids are an important type of biochemical that are building blocks of proteins. Then we need to know about a bit about DNA replication. So DNA replication is the process of copying DNA, that is producing multiple copies of DNA. Then we have transcription. It is the first step required to produce a protein. In this step, the nucleotide sequence of a gene from one of the DNA strands are transcribed, that is copied into a complementary molecule of RNA. What is translation? Translation is the process in which the nucleotide sequence of an mRNA molecule is translated into a chain of amino acids to form a polypeptide that will consist in part or totality of the protein. Then we have exons and introns. In eukaryotes, the transcribed region of a gene consists of a combination of coding segments called exons interspersed with non-coding segments called introns. Then we need to know a bit about polymorphism. So polymorphism talks about point mutations that consist in the change of one nucleotide by another are the most frequent type of genomic alteration. So when they occur with a certain frequency, for instance, in more than 1% of the population, they are called polymorphism. So single nucleotide polymorphisms also occur, which is SNP for short, are variations of a single nucleotide in the DNA sequence among a group of individuals. Then we have indels, which are other forms of variation, which means the insertion or deletion of a small number, typically less than a dozen, of nucleotides, generally called indels. And we need to know a bit about central dogma of molecular biology, which you need to study separately. I'm not gonna explain much about it. So basically, DNA and RNA and proteins are the central element of this flow of genetic information that occurs in two steps, mainly transcription and translation, in what is also called the central dogma of molecular and cell biology. So let us dive deep into biological data. So biological data mainly consist of nucleotide sequences, protein structure, and gene expression profiles. So these are a very important element in bioinformatics, because in bioinformatics we deal with biological data. So nucleotide sequences of DNA or RNA which determine genetic information. These are called nucleotide sequences. So these are basically the base pairs: adenine, thymine, cytosine, guanine. We get this type of data when we sequence information in lab, for example, for metagenomic sequencing or uh, other kinds of sequencing like 16s RNA sequencing or 18s rRNA sequencing. So there are other forms of data like protein structures. So mainly there is the 3D shapes of protein which influence their function. So the tertiary or quaternary structure of protein. So these are basically we use those data to predict the three-dimensional structure and sometimes functions. So often we use machine learning algorithms and other algorithms to predict the structure. Also, genotype, that is the genetic composition of an organism in its genome, is can also be useful as a biological data, and also the phenotype, the set of physical and observable traits of an individual, and also gene expressions. The DNA contains the genetic information required for the cells to function and replicate, and genes, which are region found along the DNA, encodes the information necessary to synthesize proteins and other molecular products. This process of using the information encoded in a gene to produce functional gene product is called gene expression. So there is something called transcriptomic sequencing. So in those cases, we also use gene expression data. So also there are some commonly available bioinformatics tools and databases. So briefly, we can introduce the online database, or what is databases. So databases are organized data collections that bioinformaticians use to store, search, and retrieve biological information. The development of new sequencing technologies created an exponential growth of sequence data, so which has gave rise to the need to develop efficient, scalable, and consistent ways of transferring data. So there are biological sequence databases can be divided into primary and secondary databases. So there are primary databases which contain sequence data submitted by the researchers that may not be fully processed and is not curated. So in such cases, redundancy often occurs in the form of multiple entries for the sequence of a gene or transcripts submitted by different people working on related topics. Also, there is another critical issue. Since data is shared by different databases, an error or an inaccuracy in the data of the primary database may easily propagate to other databases that use data from these sources. So there are commonly available primary databases which include the European Nucleotide Archive, or the ENA database, the GenBank, and also the DNA Data Bank of Japan, which is in short known as DDBJ. So there are also secondary databases as well. They contain data that are usually obtained from primary databases and that have been curated by specialists in terms of consistency and completeness. The data entries are complemented and annotated with metadata and additional information. So there are a lot of secondary databases which include NCBI, reference RefSeq databases that has been curated from GenBank or the UniProt KB or SwissProt. So there are some popular databases like NCBI Gene, NCBI RefSeq, GenCode, NCBI Protein, UCSC Genome Browser, and also UniProt, which is an integrated and comprehensive repository on sequence and functional protein information. There is Protein Data Bank, which is a database that contains structural data of protein, nucleic acid, and other complex assemblies. It also provides functionalities for data deposit and download and tools for data visualization in multiple data formats. So it has sequence, annotations of secondary structure, and tri-dimensional 3D coordinates and views as well of protein data. Then we have also DVSNP for SNP data, and for gene expression profile we have gene expression omnibus database. So these are some examples of databases that are present online. So we will talk about more about these databases in a later video. That's all for today. Thank you all for your time, patience, and understanding.